Hello, this is Team 5. This video will examine the modulation of serotonin transporter function by kappa opioid receptor ligands.
First, let us look at the background necessary to understand the results of this study. Kappa opioid receptors, KOR, agonists may be used to treat chronic pain. KOR agonists influence a patient's psychological state, and is it important to know why? Addictive pain medications should not influence cognition. Kappa opioid agonists may intentionally be used in place of SSRIs if they are deemed effective and safe for this usage. KOR's mechanism was suspected to be a kinase cascade, as it is part of other cascades in other neurons. This study looked at adding various kinase inhibitors. If the kinase is inhibited and does not affect the SERT activity, then it was considered to be not a part of the chain. Additionally, immunoblot assays were used to see which kinases showed increased phosphorylation after a KOR agonist was added.
Kappa opioid agonists decreased serotonin transporter SERT function through several mechanisms, including SERT phosphorylation, decreased surface SERT, and decreased SERT exocytosis, all which decrease serotonin reuptake. This means that agonists have a somewhat similar mechanism to SSRIs and explain their antidepressant effects. It was already known that kappa opioid receptor agonists and antagonists influence a patient's psychological state, but the mechanism by which this happens was unclear. This mechanism could be used to develop medications that deliberately treat psychological disorders and eliminate unwanted side effects. Now we will look at a few select figures of this experiment that are the main findings of this research. Figure 1 shows that SERT-HT uptake decreases as U69594 and U50488 are added as seen in A through D. Uptake recovers if KOR is inhibited in figure E or removed in figure F. Thus, U69594 and U50488 are agonists to KOR, and KOR inhibits SERT. To determine whether KOR inhibits SERT activity via kinase inhibitor, cells co-expressing SERT and KOR were treated with kinase inhibitors, including the 5 seen here. This figure found that PD98059 is the only kinase inhibitor which did not affect SERT function. Kappa opioid antagonists produced additional inhibition of SERT function when added to PD169163, PP2, and PD98959. Figure 4 looked at how U69593 inhibits SERT. SERT only has biotinylation on the surface of the cell. MESNA in C and D removed biotinylation from outside the cell so that only biotinylation was new inside the cell to look at SERT endocytosis. Sulfo NHS acetate in E and F blocked background biotinylation and measured exocytosis of SERT. This figure found that U69593 speeds up SERT endocytosis but slows SERT exocytosis.
Based on the results of this study, researchers determined that U69594 and U50593 are agonists to KOR. KOR inhibits SERT via endocytosis. Thus, U69594 and U50593 slow SERT and HT5 uptake. These results may be useful for treating serotonin-related diseases in the future. In conclusion, this study was used to determine the mechanism of how KOR agonists decreased SERT function. This study determined that activation of KOR inhibits SERT activity through endocytosis. The mechanism of SERT inhibition is also distinct from SSRIs. These findings could be used to more deliberately treat psychological disorders, specifically serotonin-related diseases, with limited side effects in the future. Thanks for watching.